Streptococcus or Streptococcus pyogenes causes strep throat, and it's also the leading cause of flesh-eating disease worldwide. Way more than Ebola or other viruses. It literally is Streptococcus, which causes 500,000 deaths every year. But one of the reasons why it's such a pathogen is because of the viruses that infect it, or bacteriophages, right? So they carry the genes that make you sick, and when they infect a Streptococcus bacteria, they give the Streptococcus that gene, and so it becomes more virulent, right? So this infection process is responsible for the rise of new pathogenic strains of Streptococcus pyogenes constantly and every year. Many people carry Streptococcus pyogenes and group A strep on their bodies as commensals, and it doesn't make you sick. So one of the big questions in the field is like, when and how and why does it make you sick, and why does it have to be a pathogen, and the interaction between Streptococcus and the virus. And in our latest study, we discovered that it inhibits and binds tons of other proteins in Streptococcus pyogenes to control the bacteria. So this one little phage protein is what we hypothesize is helping control the relationship between the phage and the bacteria. So, and what part of the process to maintain the phage genome and to keep Streptococcus as a pathogen. So our publication that just came out basically showed we had discovered that it bound other proteins before, and now we say, hey, look, it binds all these different DNA regulatory proteins of the bacteria and the phage. So it's using this one protein to just control the entire metabolism of Streptococcus pyogenes. So I think everyone's had strep throat at some point in their lives, and that's this phage protein is part of this regulatory cycle and what we think is controlling the pathogenesis of Streptococcus, keeping Streptococcus a pathogen. Flesh-eating disease worldwide, again, is caused by Streptococcus. So if we can understand this relationship and understand why and how the mechanisms of why this bacteria that doesn't have to be a pathogen becomes a pathogen, maybe that's an alternative, not just to understand what's going on, but maybe if we can control this process and understand these mechanisms, we can keep Streptococcus being a beneficial bacteria and not being a pathogen. What's neat about this project is it just keeps giving and it keeps opening up more doors. We've discovered that it binds a whole bunch of other proteins, but many of these proteins are in regulatory pathways that control the biology of Streptococcus in ways that are completely unstudied and not understood at all. But now we have to characterize all these brand new proteins and brand new pathways and understand how they affect the biology and metabolism of Streptococcus. What's cool about it, it's often people are like one protein, one function, or one protein, a set amount of functions. But every now and again, you find a protein like this, which has multiple functions. At the very least, it affects 5 or 6 different proteins that are all in 5 or 6 different pathways that are important for controlling the metabolism of Streptococcus. This is the first time we've published nucleic acids research. I was really excited. Nucleic acids research is one of the top tier journals in biochemistry, and it was a really good fit for our paper because the journal really focuses on understanding the biochemistry of nucleic acids such as RNA and DNA, but also all the proteins that manipulate RNA and DNA and interact with them, right? And so for our system, all the proteins that are controlled and interacted with by our one little phage protein are all DNA processing proteins. So they regulate how Streptococcus takes in new DNA, they regulate how Streptococcus communicates with itself to take in new DNA. What we found now is it also binds all these extra proteins involved in processing that DNA. So it was a great fit to understand these proteins and this new discovery of all these different interactions and metabolic pathways. The credit for this really goes to Muna, the first author, and who graduated with her master's, and then Nicole, who graduated with a master's a couple of years ago, that got a lot of the preliminary data for this, like figured out that it does bind other proteins, and then Muna came in and worked out a lot of the initial biochemistry and solved the structure, so they deserve all the credit. And then what was really nice is Julia was a summer student of mine for a bit and then worked in the lab under Muna and then made a bunch of the constructs that we use and helped purify proteins. It was her first publication as an undergrad, which was really cool. And since all these proteins are unknown or are unstudied, we get to name them, which is nice because they just have gene names, and usually then you kind of rename it based on a function. So it's a rare instance where Muna and Julia have a protein named after them. So one of the proteins in our studies is JM3, which stands for Julia Muna Construct 3. So they have proteins named after them. Then we still have to figure out what it does, but it's cool to have a protein after them. So on that note, it's really important for me. Everyone needs to feel that they're involved and have control in the lab in some way, so that they have control in their projects. There's an open channel with me, a lot of freedom to explore, but then always being around to help. They can ask questions. I'm in the lab. We have regular meetings. And then every decision for the lab is a joint decision. I'm the executor where like things are the final, and then we decide all those together. So really being very interactive and just being there for your students too. So one of the other exciting things about this project, because we have one protein that binds a lot of other different proteins, is that one of my collaborators in the Department of Chemistry, Dr. Mazdak Kajapur, is also interested in this protein and how it folds. What we've discovered is that this protein is also something called an intrinsically disordered protein, which means it normally has no fold and it's just wiggly until it finds its binding partners, and then it adopts a fold. And what we found, or what's hinting at, is like it looks like it may be changing its shape depending on who it may bind, right? Which is also a really cool discovery and a rare thing that you find just in protein science in general, but that has yet to be proven. But because it has so many different binding partners that have different surfaces, it's a hypothesis that we're currently pursuing.